Okay, we're going to get started, and if you want to sign up for the course, this is the last day, and as many of you know, there's a potential government shutdown tomorrow, so we won't be assigning any course until the government is fully in action. And also, if there is a shutdown and it lags on to next Monday, well, if there's no the government is closed, TRACO is closed. So we'll hope that if there is a shutdown, it's a very short one. So our first speaker today is Christina Annunziata. She got an MD from Georgetown University Medical School, and then she came to NIH and worked with Lou Stout on the NF-kappa B pathway in multiple myeloma. And now she's a principal investigator working on ovarian cancer. The title of her talk, Ovarian Cancer in the Genomics Era. Tina.
Okay. All right. Thank you very much. What is this? Do you put up my slides or do I put up my own slides? You will get them. Okay. Well, thank you for having me give this talk. I've done this for a couple of years now, but I just kind of update the slides every year. So we should see a little bit more. I used to talk about angiogenesis in ovarian cancer, and now I'm talking about genomics, so that's a little bit more modern, although angiogenesis is still an interesting target. So I was first going to start talking about ovarian cancer from a clinical standpoint, so we can sort of have some background. Are we up yet? I'm not touching anything. Okay, here we go. Okay, great. All right, so ovarian cancer can be defined as 4 stages, as most cancers can be. Stage 1 of ovarian cancer is confined to the ovaries. Stage 2 is confined to the pelvis. Stage 3 is spread beyond the pelvis into the peritoneum or the intraperitoneal nodes. And then stage 4 is distant solid metastasis. So as you can see, should I use this pointer here? Most of the ovarian cancer is diagnosed in a late stage of disease, and so that's why the survival is so poor in ovarian cancer. If ovarian cancer can be detected in early stage, survival would be much better, and this is sort of where we get diagnosed in breast cancer. Most of the breast cancers are stage 1 and stage 2, as opposed to ovarian cancer, which is more diagnosed in stage 3 and stage 4 disease, where the survival is much worse. Other prognostic factors other than the stage for ovarian cancer is the extent of cytoreduction, and that's the amount that can be taken out during surgery, and we'll talk about that in a little bit more depth later. The histology of the ovarian cancer, it can be mostly serous histology, but the clear cell histology tends to portend a worse prognosis. And then the grade, of course, if it's higher grade, which means more proliferative activity, worse, more undifferentiated morphology of the cells, that also portends a worse prognosis. The performance status is a measure of how well the patient is doing. So if the woman with ovarian cancer is in poor health at the time of diagnosis, that she'll have a worse outcome as opposed to a woman who is in good health. P53 status, although this may not be an independent predictor because it's very closely tied to the histology. Vital organ function is related to performance status and physiological age. Also, we're going to talk about platinum and taxane as primary therapy, that if people can get all 6 cycles of their primary therapy, that's a better outcome. Intraperitoneal therapy we'll talk about, and BRCA and VEGF production we'll talk about as well. So what is the treatment for newly diagnosed ovarian cancer? The first step, of course, is complete surgical staging. The next is optimal reduction of the tumors based on surgery. The next would be chemotherapy, and then, of course, clinical trials. So complete surgical staging. This involves full assessment of the abdomen with random biopsies, even of visually negative areas, and that's to make sure there's no microscopic disease in areas outside the pelvis or in abdominal organs. Lymph node dissection should be done except for it's clearly stage 1, but of course it cannot be staged as stage 1 if the random sampling has not been done. So where does ovarian cancer go? So you can imagine it starts first in the ovary here. Of course, there's a newer literature that says it could start in the distal fallopian tube, but let's just say for purposes of this picture that it starts in the ovary. Where does it go next? So the ovarian cancer does not spread primarily by lymph nodes or blood, such as in breast cancer or other solid tumors. The first spread of ovarian cancer is through the ascites, which is the peritoneal fluid. So it's the fluid that's circulating and bathing the abdominal organs. So the first place it goes is it kind of just releases off the surface of the ovary as opposed to going into the bloodstream and having to invade that way. And the way it goes is that it goes into the peritoneal fluid and circulates in this sort of clockwise direction, because that's the typical way of circulating the peritoneal fluid. So you can imagine if the cells float off of the ovary, they're going to go here, and the first place they'll stick is actually on the top or the outside of the liver and stick in this, like in between the liver and the diaphragm. So if it's stuck right on the outside of there, and not invading, it's considered a stage 3 because it hasn't invaded the liver. It's still considered sitting on the outside of the peritoneum, which is the tissue that coats that covers the abdominal organs. It has not invaded. If it were to invade the liver, then it would be a stage 4. So once the surgery has been, once the patient has been staged, then the next step is in that same operation to perform as much cytoreduction as possible. In stage 1 and stage 2, of course, the goal is to remove all of the disease. In stage 3 and 4, the less disease you leave, the better. And of course, what's considered optimal nowadays is less than 1 centimeter, but that's probably going to change and be actually no visible disease. And that's based on this. So this is a historical graph of survival, long-term survival, just based on the amount of surgical cytoreduction that could have been done. So you can see here, if patients were, and this is a very old, this is a very old slide, this is like from 20 years ago. If patients were left with 2 centimeters of disease, greater than 2 centimeters of disease, their outcome was much worse than if they were left with no visible disease at the time of finishing up the surgery. If it was 0 to 1, that was better than if there's 2, but still not as good as 0. So as of now, optimal cytoreduction is considered less than 1 centimeter, so it will be kind of like on this half as opposed to this half. But like I said, that's changing because surgeons are becoming more and more aggressive, and we're getting better at getting 0 centimeters left. This is kind of tricky because here you can have, it's 1 centimeter, not total disease left. It's 1 centimeter, like the largest blob left is 1 centimeter. So if you have a sheet of tumors on your peritoneum, that's still considered optimal debulking. So that's why, that's why the survival is so much worse than if you had nothing left. Now, this is also a question of, is it the type of tumor, is it the biology of the tumor that's different? So this is also up for discussion. If you can get everything, is that sort of a different type of biology? So for example, if it grows like in individual nodules, is that a different type of tumor than if it's growing in sheets and you can't get everything? So that's up for discussion. But just to make the point, if you can leave nothing, you have a better prognosis. So after surgery, the next step would be to give the patient chemotherapy, and the standard chemotherapy is a platinum and a taxane. And that means usually it's carboplatin or cisplatin in combination with paclitaxel or docetaxel. So that is standard. The way you give it is up for discussion. And so there's a lot of trials going on right now to decide how you do it, and I will show you a couple of those. So that's just the history of this. The survival of ovarian cancer actually was markedly improved with the addition of platinum in the 70s. Before the 1970s, no one with ovarian cancer lived 5 years. In the 1970s, platinum agents were introduced, and we got 5% of the people to live for more than 5 years. Then, with the addition of the taxanes, so with paclitaxel was the first taxane, the survival jumped to 35%. So that's actually a big improvement in survival here. Since then, we have gone slightly better, and this intraperitoneal is actually not a different type of agent, but it's a different way of giving the chemotherapy. So instead of giving the chemotherapy IV, they're given both IV and directly into the abdomen right after surgery. That had a significant improvement in overall survival at 5 years, but not a huge jump like with the addition of the taxanes. Other things have been, other agents, chemotherapy agents have been combined with platinum, and none has been actually better than combining with paclitaxel. The thought is that this is kind of a key thing to notice here. The paclitaxel actually spares the platelets. So all these other things in combination require dose reductions of the chemotherapy because they add to the toxicity of the platinum, which is most commonly your blood counts going down. So red blood cells, white blood cells, and platelets going down. But for some reason, the paclitaxel actually spares the platelets. And so the doses of both the carboplatin and the paclitaxel are able to be maintained at full dose because of that. So the new standard of care, and I say that with a grain of salt because it's not completely implemented at the moment, the new standard of care is the intraperitoneal therapy based on this trial, which was done by the Gynecologic Oncology Group, which is a cooperative group run by or sponsored by the National Cancer Institute. This was a multi-center clinical trial testing the standard therapy with intravenous cisplatin and paclitaxel given in comparison to intraperitoneal therapy. So the cisplatin, interestingly, was given just intraperitoneally because that gets absorbed into the circulation by nearly 100%. However, the paclitaxel was given both intraperitoneally and IV because the paclitaxel is in a type of hydrophobic formulation which does not permit transversing of the peritoneal membrane. So paclitaxel, when you give it intraperitoneally, does not get into the circulation. So it had to be given IV as well. So you can see the total dose that you're giving the patient is actually higher in this arm. Only 42% of patients were able to complete all 6 cycles of therapy. However, there's still, even with that caveat, there still was a dramatic improvement in overall survival. This was published in the New England Journal in 2006, where this median survival of the standard arm, which was just the intravenous, was 49 months, whereas the overall survival of the intraperitoneal chemotherapy arm was over 5 years, so it was 65.6 months. So that's a dramatic increase in overall survival. Now, when I say that we take this with a grain of salt, is that this was actually only included patients with stage 3 disease, so not stage 4. So it's not clear whether patients with stage 4 disease will benefit from this therapy. The patients had to have optimal cytoreduction. So if you remember that I showed you before, optimal means less than 1 centimeter of disease remaining. So you must fall into that category of stage 3 optimal cytoreduction to actually have qualified for this trial, for this to be administered. The other caveat is that it is actually a very difficult regimen to tolerate. As I mentioned, only 42% of the patients on the study completed all 6 cycles. So it's not very well received by women with even the perfect amount of disease and the perfect scenario, it's still not very well received. So it's not been widely implemented as the new standard of care, even though this was shown to have a dramatic improvement in overall survival. So where do we go from here? So what about genomics of ovarian cancer? So how can we improve on this 5-year median overall survival with intraperitoneal chemotherapy? So this is the translational oncology course. So what is translational oncology? We start with an observation, get a hypothesis, can do an experiment or a clinical trial, get a result, analyze the result, and come up with a new hypothesis. So we're going to try to use genomics to go through this scenario. In 2011, the Cancer Genome Atlas published their comprehensive genomic analysis of ovarian cancer in Nature. The background here is that the Cancer Genome Atlas had nearly 500 clinically annotated high-grade serous. So this is what HGS stands for, high-grade serous. That's the serous subtype of ovarian cancer. They had nearly 500 samples where they identified molecular abnormalities that would influence pathophysiology, affect outcome, and constitute therapeutic targets. This was the goal of the genomic analysis. So what did they do? They looked at gene expression, so messenger RNA expression. They looked at microRNA expression. They looked at DNA copy number. They looked at DNA promoter methylation, and they also sequenced whole exome sequencing in 316 of those samples. The inclusion criteria was that they had to be newly diagnosed patients. They had to have serous subtype of ovarian cancer, no prior treatment. They could have any surgical stage or grade. No, they had to be high grade, sorry. They could have any surgical stage. They had to have frozen tumor specimens with a companion normal tumor specimen. In most cases, the companion normal tumor specimen was peripheral blood DNA and not normal ovarian surface epithelium. Although there were a few cases where they had normal ovarian serous epithelium from the non-involved ovary. The clinical data actually was accessed, so this means how long the patients have survived, whether they were resistant to chemotherapy initially, whether they responded to chemotherapy and then recurred. Also the demographics of the patients. All of this can be accessed through the website. Actually, it's not this website anymore. It's cancergenome.gov is the website. They have demographics, they have the pathologic information, they have treatment details and outcome parameters from these ladies. So it was a very complex analysis because there were multiple centers involved with the Cancer Genome Atlas, and this probably applies to the other histologies of cancer, the other types of cancers that were included in the Cancer Genome Atlas as well. So first there was a preliminary pathology review to sort of find the cases. They started out with 1,020 cases here. They did a pathology review, and actually they took out 200 cases here. 200 cases failed the pathology review. They went through analysis of the quality of the specimens and took out another 243 that were poor quality. And then they also collected the clinical data elements of these. And then they sent them to 7 characterization centers, the data coordinating center and 3 sequencing centers. These were 518 cases here, but then additional 46, which have been subsequently included, are not included on the initial analysis. So it was a very large effort on the part of the Cancer Genome Atlas for ovarian cancer as well as for the other cancers. So what did we find? So they found, not we, I was not involved, with the ovarian cancer specimens, specifically with the sequencing. This is in the 316 samples that were sequenced. Of course, the highest mutation was found, the highest rate of mutation was found in the p53 gene. So now, based on this, I mean, nearly all of them had a p53 mutation or a loss of p53. So now high-grade serous ovarian cancers are sort of thought to be kind of universally dependent on the p53 mutation. Another 10%, excuse me, almost 10% had a BRCA 1 or 2 mutation. And then these other genes that were found to be mutated in a relatively high frequency. But still, I mean, there's not a lot, there's nothing really that jumps out at you other than the p53 and the BRCA, and then maybe these other ones in here, and the RB and this one are fairly infrequent. So there's not a high amount of frequency of mutations in high-grade serous ovarian cancer. And we'll get to that in a second, which is right here, which is why. So if you can see here, so in the glioblastoma, this is comparing the DNA copy number. Here on this axis here are the individual chromosomes. Blue is if it's deleted, white is it's neutral, and red is it's amplified. So you can see here in glioblastoma, which is actually the first tumor that was sequenced and that was analyzed through the Cancer Genome Atlas. In glioblastoma, there's very distinct areas of amplification, distinct areas of deletion, and then most of the other parts of the genome are relatively unaffected. As in contrast to the ovarian cancer, where pretty much the whole genome is affected. There's not really one area that's particularly white. Everything is either blue or red. And so this is showing you that there's just a high level of DNA copy number variation throughout the chromosomes, throughout the whole genome of serous ovarian cancer. There were molecular subtypes identified by gene expression profiling. And this was a reference that was published a couple years before in 2008, where they had 245 tumors. Here are the 489 tumors. They used 1,000 genes to subgroup into 4 different subgroups based on sort of the genes that were, the networks that were found in those subgroups. So one of the subgroups they called differentiated, one of them was immunoreactive, one of them was mesenchymal, and one of them was proliferative. They tried to say that, okay, that we validated this in another related data set, which is the 245 tumors in reference 25, that there was a similar breakdown of tumors in these 4 categories. So what does that mean? So putting this together, I'm jumping a lot again. This was a very detailed paper published in Nature, so I'm really just skipping to the highlights here. But what does this mean? So they're saying, well, we have a high level of genomic chaos. We have a lot of deletions. We have a lot of amplifications. We also have some overexpression. We have some underexpression. How can we put this all together? So they tried to group it into pathways that were altered. So what pathways did they found? They found that there was 67% of cases had some alteration in the RB signaling pathway, and this is broadly defined as cyclin CDKN2A, cyclin E, which is CCNE, cyclin D1, cyclin D2, and RB. And so you could see that all of these kind of aberrations would lead to cell cycle progression, with these being downregulated, the inhibitors being downregulated, and the activators being upregulated. Another 45% of cases had alterations in the PI3 kinase and RAS pathways, and that's defined by things, again, either being deleted, amplified, or mutated in this case, with very rare mutations in BRAF. Notch signaling came out as another pathway in 22% of cases with amplification or deletion, mostly amplification or mutation of cases of genes in this pathway. The most common thing that was altered was probably the homologous recombination. So here is what they're calling homologous recombination alterations, either BRCA1 germline mutation in purple, somatic mutation in green, or epigenetic silencing, meaning methylation of the gene, in this blue and gray box. So in these cases, in 33% of the cases, they're saying there might be a defect in DNA damage repair, in the DNA damage repair pathway. And this was the homologous recombination pathway. I'm going to talk about this in a little bit more detail later. But the thought was that 51% of the cases have this alteration in this pathway. And actually, the ones with the BRCA mutations had a better survival than ones with epigenetic silencing or wild type. So this might get to the question of what we saw at the very beginning, which was that all of the cases had these crazy genomic patterns of genomic amplifications and deletions, and nothing really that specific, but kind of whole genome, might be because there's defects in DNA repair. So this is kind of what came out of the TCGA, is that number one, p53 is mutated, and number two is that there is some global defect in repairing DNA. Again, these are pathways. This is kind of another way of representing it, that the DNA repair pathway was altered and cell cycle progression pathways were also altered. So again, just to summarize, the TCGA was a large-scale integrative view of aberrations in high-grade serous ovarian cancer. The mutational spectrum was emphasized as being surprisingly simple, where p53 mutations were in 96% of the cases, BRCA1 and BRCA2 were in about 22% of the cases, and there were 7 other significantly mutated genes in about 2 to 6% of cases. This makes high-grade serous ovarian cancer distinct from the other histologic subtypes of ovarian cancer as been published in the literature. So for example, there's a subtype of ovarian cancer called clear cell histology, where there's very few p53 mutations. There's recurrent ARID1A mutations, and there's recurrent PI3 kinase mutations. Similarly, in the endometrioid subtype of ovarian cancer, there's frequent beta-catenin, ARID1A, and PI3 kinase mutations, and very few p53 again. And then in the mucinous subtype, there are prevalent KRAS mutations, making that sort of a targetable pathway in mucinous ovarian cancer, and again, distinct from high-grade serous ovarian cancer. So again, to summarize, there was a remarkable degree, this is quotations from the actual manuscript in Nature, remarkable degree of genomic disarray, striking contrast to previous TCGA findings in glioblastoma, mutations and promoter methylation in putative DNA repair homologous recombination may explain the high prevalence of copy number alterations. So what next? So how can we use this information to try to improve the overall survival of women with high-grade serous or with any type of ovarian cancer? 50% of the patients were thought to have homologous recombination defects, and there have been drugs developed called PARP inhibitors. I'm going to talk about that in a minute. PARP inhibitors might be better at targeting women with, or cancers with defects in DNA repair. There were also commonly mutated pathways such as RB, RAS, PI3 kinase, FOXM1, and Notch. So perhaps we can find molecules that target these pathways specifically and give those to women who show aberrations in those type of signaling pathways. And they did mention there are inhibitors for 22 genes that are in regions of recurrent amplification. I think the theme here is that not one gene can be targeted, but we probably need to target networks and direct the therapies towards the networks in order to have a better improvement in survival. So now we get to clinical trials. So what are we going to use for targeted therapies in individual clinical trials? And you can imagine that one of the things that we could use is a PARP inhibitor. So actually, this predated Cancer Genome Atlas, because in 2009, there was a discussion at the ASCO, which is the American Society of Clinical Oncology annual meeting, that discussed a phase 2 trial of the oral PARP inhibitor called olaparib in BRCA deficient advanced ovarian cancer. So what are PARP inhibitors? So we need to understand the pathway of DNA repair. This is a very simplistic schematic diagram of DNA repair, but I'm going to put it in context for the PARP inhibitor. So with normal cellular metabolism and environmental exposures, there can be single-stranded breaks delivered to the DNA that need to be repaired. PARP is one of the enzymes that identifies and sort of highlights single-strand DNA breaks, and the way it does that is the PARP stands for poly-ADP ribose polymerase, which is an enzyme that binds to these single-strand breaks, recruits other elements, and puts polymers of ADP ribose onto there to sort of flag it. In replicating cells, if the single-strand break is not repaired, that single-strand break at a replication fork will be converted into a double-strand break because the replication machinery won't be able to get through there. It'll make the break, but it won't be able to repair the rest of the strand. So if you have a PARP inhibitor, in a normal cell, this double-strand break will get repaired by a pathway called homologous recombination. So in homologous recombination requires the BRCA proteins in order to successfully complete. So if the BRCA proteins are there, homologous recombination will ensue and you will have the cell repaired and surviving. However, in cells with a BRCA deficiency, there is no repair of this. There are other repair mechanisms, but they're not as efficient and they're much more error prone. So if the single strand breaks, if the double strand machinery gets overwhelmed, the cell will not be able to repair the DNA and the cells will undergo cell death. So olaparib is an orally available PARP inhibitor that was shown in cell culture, at least, to have synthetic lethality in homozygous BRCA mutant cell lines. So the primary aim of this clinical trial was to test the efficacy of olaparib in confirmed BRCA1 or 2 mutation carriers with advanced refractory ovarian cancer. They enrolled 57 patients. 39 of them had BRCA1 mutation and 18 had BRCA2. There were 33 evaluable at a dose of 400 milligrams twice a day and 24 at a dose of 100 milligrams twice a day. The overall response rate was higher in the 400 milligram arm, as expected. However, the clinical benefit rate clinical benefit rate was defined by response, so shrinkage of the tumor, or a 50% decline in a tumor marker that we use for ovarian cancer called CA125. Of course, again, there was a higher clinical benefit rate at 400 milligrams as opposed to at 100 milligrams. Progression-free survival was 5.8 months, and the median duration of that was 9.6 months. The toxicity was actually quite similar to chemotherapy in that grade 1 or 2 nausea, so that's mild nausea, fatigue, anemia, and low white blood cells, which is leukopenia. This is a waterfall plot that shows you the best response of each patient on the clinical trial. So this means that the tumor grew, and then below 0, it means that the tumor shrunk. So in actually in 3 patients, there was a complete clinical response. So this is actually quite good, because in recurrent refractory ovarian cancer, we rarely get complete responses, and a lot of these women had multiple lines of chemotherapy prior. So this was actually quite remarkable that most of the patients had some sort of shrinkage, and actually some of them even had complete resolution of the tumor. This is a graphical representation of what I already mentioned, which is that the 400 milligram dose did better than the 100 milligram dose. So the conclusions from this in 2009 was that oral olaparib was well tolerated, highly active in advanced chemotherapy refractory BRCA deficient ovarian cancer. Greater activity, of course, at the higher dose. Toxicity was similar to that seen in non-carriers. That's not a direct outcome of this trial, but that's compared to previous trials. And they quoted a positive proof of the concept of the activity and tolerability of genetically defined targeted therapy with olaparib in BRCA deficient ovarian cancers. So this is actually quite a big step. This is now being tested in a randomized placebo-controlled clinical trial for women actually in first remission of ovarian cancer to see if we can get that overall survival bumped out beyond 5 years. So now I'm going to tell you a little bit about where we've been going in the lab here. We're looking at NF-kappa B deregulation and targeting. The other olaparib story kind of took us from the genomic aberration to the pathway alteration. And from this, we're taking a different approach, looking from the pathway and looking for genetic alterations that we can find that might highlight women that have aberrations in this pathway. So this is a highlight, just a schematic to show you a very simplistic view of the NF-kappa B pathway. This is in just a generic, not specific to ovarian cancer. But you can see that it's far more complex than this, but you can start with TNF signaling. You have activation of this heterotrimeric complex of IKK alpha, IKK beta, and IKK gamma, which then activates the NF-kappa B proteins, which are actually transcription factors. I-kappa B alpha, which is the inhibitor of kappa B alpha, needs to be phosphorylated and degraded. And then these transcription factors, the NF-kappa B transcription factors, usually function in dimers of heterodimers of either P65 and P50, or another components could be P52 and REL-B. And then once those are released, they can travel into the nucleus, bind to the DNA, and promote transcription of target genes. One of the key functions here is that there's a molecule called CIAP, and that stands for inhibitor of apoptosis. That can be involved in activating NF-kappa B by promoting the heterotrimeric complex to phosphorylate and degrade I-kappa B alpha. And it's also important, obviously by its name, in inhibiting apoptosis. So we started out just looking at to define ovarian cancer NF-kappa B signaling, and we started by using an IKK beta inhibitor. We wanted to define an IKK beta or an NF-kappa B gene expression signature so that we could go and look in large gene expression databases of ovarian cancer and to see whether this was an important pathway or whether we could find if it had a survival difference or how often it was expressed. So we used either an IKK beta inhibitor, which was a drug, or we used an IKK beta shRNA to knock down IKK beta in order to develop, to look for genes that were consistently downregulated across both drug and shRNA in order to define a signature to probe larger data sets. So we found that these 9 genes were consistently deregulated upon treatment with either the inhibitor or the shRNA. So we define this as our 9-gene IKK beta signature in order to use in further studies. So we looked in a bunch of data sets, actually this is just one of them, showing that these genes actually are co-regulated, where red is overexpressed and green is underexpressed. And this is a set of 185 ovarian cancer samples. Each column represents an individual patient, and then each row is a particular gene in the pathway. So what you can see, just kind of looking at this as an overview, there are a set of patients which have high coordinated expression of these genes. Here's the correlation with each gene with its average, and this is a very highly significant correlation across these 185 genes. I didn't include the p-value here, but they're all less than 0.0001. So what this tells you is that because they're coordinated, it suggests a biological function of these genes not just random, some are up, some are down in various cases. When they're up, they're all up. When they're down, they're all down. So that implies that there is a biological significance linking these genes together. And we did verify these in other data sets as well. So when we looked at these genes in overall survival, there was a significant, not a huge difference, but there was a significant difference if the patients with the very high expression of the 9 genes had a worse overall survival than those with very low expression of the 9 genes. So we wanted to find out, okay, we found out, we convinced ourselves that NF-kappa B is active in ovarian cancer. There are patients who have a high level of NF-kappa B activity, and therefore, perhaps if we block NF-kappa B signaling in these patients, this might be a good therapeutic target for these patients. But we wanted to find out, excuse me, how can we target it, and how could we, and what other pathways we would be affecting. How is NF-kappa B actually turned on in ovarian cancer? So what we did was a shRNA library screen with a shRNA library that's targeting 500 kinases in the human genome. We combined the shRNA screen with or without a IKK-beta inhibitor in order to see which pathways actually would enhance specifically the effect of the IKK-beta inhibitor without, while not looking at things that were just broadly toxic across all ovarian cancer cells. Excuse me. So the way this works is that the shRNAs were transfected into a cell line. They are bar-coded, so each of them can be detected at the end by its barcode. It's a 6-dimer barcode. It was infected into the cancer cells. We induced the shRNA in all the cells, and then treated half of them with the IKK-beta inhibitor and half of them with just the vehicle. And what we were looking for in the readout were shRNAs that were lost only in the IKK-beta group, in the inhibitor-treated group, and not in the IKK-beta negative group. Excuse me, in the inhibitor negative group, in the vehicle control group. And if it was lost in both equally, we didn't look at that at this stage. We were just looking for things that enhanced the toxicity of the IKK-beta inhibitor. And what we found actually was that the gene caspase-8, when that was knocked down, that actually was specifically toxic to cells that were treated with the IKK-beta inhibitor. So how can we use caspase-8 to help to try to treat ovarian cancer? So what I had told you about that the NF-kappa B pathway, specifically with IKK-beta, requires this signaling through the inhibitor of apoptosis, or the IAP molecule. Now, in normal cells, there is a protein released from the mitochondrion to promote apoptosis, which is called SMAC, which stands for Second Mitochondrial Activator of Caspases. So when SMAC is released from the mitochondrion, it actually binds to IAP and gets rid of IAP, and that main function is to allow signaling to activate the apoptosis pathway. But then also what happens when you get rid of IAP, you also shut down NF-kappa B signaling. So where does caspase-8 sit in this? Caspase-8 is actually activated here during this pathway in the pro-apoptotic arm. In tumor cells, it's thought that apoptosis is dysregulated due to low levels of SMAC or to overexpression of IAPs. So this might be a pathway that we could target in ovarian cancer, and we thought we would look first in situations where there was high immunohistochemical expression of TNF-alpha in the ovarian tumor and stroma, increased TNF-alpha signaling in serum concentrations, and TNF-alpha, we thought, might activate NF-kappa B in the presence of IAPs, but trigger apoptosis in its absence. So our new hypothesis moving forward from this point was that intersection of the NF-kappa B and caspase pathways provides a novel point of therapeutic intervention where pro-survival signals can be switched to pro-apoptotic signals to kill cancer cells. Luckily, there is a SMACmimetic, so there's a drug that mimics this protein SMAC that's released from the mitochondrion. And the SMACmimetic binds to the IAPs and causes their degradation. So we thought we would look to see in a panel of ovarian cancer cell lines whether this correlated with high NF-kappa B activity. And it did. The blue lines are ones that have high NF-kappa B activity, specifically CAOV3, which was the line that we used for the library screen. So CAOV3, this line, which has high dependence on NF-kappa B signaling, was the most sensitive to this SMACmimetic. Other cell lines were less sensitive to treatment with the SMACmimetic, either by itself, and then we also enhanced the killing actually by adding TNF. So TNF can stimulate NF-kappa B signaling in ovarian cancer and other cancers, but it can also stimulate apoptosis. So what we found was that 2 additional cell lines became sensitive to the SMACmimetic killing when we turned on the signaling through the TNF pathway. And this was at a very, very low concentration of TNF, which did not kill the cells just by adding itself. We just did a preliminary look at the proteins as what's happening here, and you can see, of course, the CIAP was completely downregulated or degraded after SMACmimetic treatment. Caspase-3, which is a pro-apoptotic protein, the cleavage of caspase-3 was up. Caspase-8, interestingly, went up. And I-kappa B phosphorylation, which is this upper band, actually did go away with both an IKK-beta inhibitor and with the SMACmimetic. So where are we now? We are going ahead with a clinical trial with the SMACmimetic for relapsed ovarian cancer. It's a drug that's given weekly for 3 weeks out of 4. So this is highlighting the drug administration. And what we're doing to confirm the activity of the drug, we are doing biopsies. So we take a biopsy of the patient's tumor before we treat the patient, and then we give the patient 2 cycles of the chemotherapy, of the SMACmimetic, and take another biopsy to look at whether the IAP is degraded, number 1, and then also to look at whether we turned off the NF-kappa B pathway and turned on the pro-apoptotic pathways. And then from a clinical standpoint, we do CAT scan imaging after every 2 cycles. Our primary endpoint of this trial is objective response or progression-free survival. But we also have several correlative studies, meaning the translational experiments on the paired pre- and post-treatment biopsies. And we're also collecting peripheral blood as well and serum cytokines. So we'll be looking at the immunohistochemistry to measure TNF-alpha and TRACO. We'll also be looking at gene expression profiling to look at that 9-gene NF-kappa B signature to see if that was altered by the SMACmimetic. We're going to look at the apoptosis signature, and then we're also going to look at a lot of proteins in the biopsies. Of course, like I mentioned, the CIAP1 and 2, caspase-8, and PARP cleavage, as well as caspase cleavage. So again, we made an observation in our patients that there were potentially a group that had high NF-kappa B activity. We came up with the hypothesis that they might benefit from inhibitors of NF-kappa B. We are currently in the process of doing, well, we did a couple of experiments. We're currently in the process of doing the trial. And then once we get the results, we will analyze them and come up with new hypotheses. And I can give you some insight into where we are going. There are a number of drugs that can augment apoptosis when given to the cancer cells in vitro and in vivo, and these are listed here. So our thought is that we can promote apoptosis by treating the patients with a pro-apoptotic agent, and then also give them the SMACmimetic to augment the response to the SMACmimetic. So what we're doing in the lab right now is actually combining it with docetaxel, which is one of those drugs that can change the mitochondrial membrane potential and promote apoptosis through release of cytochrome C. So our thought is that perhaps we could prime the cells by giving them the docetaxel first and then give them the SMACmimetic. But we're going to compare either the SMACmimetic first and then the docetaxel, or vice versa, to see if we can look at proteomic endpoints and efficacy endpoints, first in the cell line and then moving to clinical trials. So our goal for individualized therapy is that therapeutic agents can target each critical pathway and they can be tested for their ability to overcome molecularly defined cancers. So I really think that this is the way we're going. We know that ovarian cancer is extremely heterogeneous. I don't think that anyone believes that any one agent is going to be a good magic bullet for all patients with ovarian cancer. So I think what we really need to move towards is defining subsets of ovarian cancer, even within the histologic subtype, to see if we can find evidence of pathway activation, pathway addiction, pathway dependence, target that pathway in that specific group of patients without having to expose everyone to a particular drug in order to get it, to get it into the clinic. Ultimately, I think the benefit of signature-directed therapy will be assessed by prospectively profiling individual patients and allocating therapy based on the molecular defects that we identify at the bench. This is mainly the clinical team that I work with. In the lab, we have excellent scientists, Elijah Hernandez, Marianne Kim, and Carrie House working on this from a laboratory standpoint. And with that, I will finish and take any questions. Yes?
For the phase 2 study by SMAC, how to choose the patient, whether they already are done some kind of like resistance towards some drug, then you put this one, how is it?
Yes. So our trial is specifically for women who have shown resistance to platinum therapies. The reason for that is because we think that this pathway might be able to overcome the resistance to platinum, because a lot of times the platinum might be due to insufficient apoptosis. We have an upcoming trial that's going to be in platinum-sensitive women that's in combination with other agents. And then we're going to do the docetaxel in combination with SMACmimetic. And that could be for anyone, platinum-sensitive or platinum-resistant.
Is there any relation with BRCA1 or BRCA2?
So that's an interesting question. There has not been anything in the lab that's shown, that BRCA mutation patients are more sensitive. There have been in the clinic, there's like 3 or 4 SMACmimetics in the clinic, and in one of them they did show that it's coming out that there is more sensitivity in the BRCA patients. Now, whether or not that's just because those cells are just generally primed for apoptosis, that I don't know. So I think that's still up for questions. Not particularly, no, they have not shown, not particularly. I mean, in general, the cell lines are kind of messed up, as cell lines are messed up, they just have like polyploidy, aneuploidy. So from that sense, there's a DNA repair defect, but not specifically like in the BRCA pathways. Okay, thank you. Okay.
Okay, so our next speaker is Sonia Jakowlew, and Sonia got a PhD at Rutgers University. Subsequently, she did a postdoctoral fellowship in France, and then she came to NCI, and she's been here, wow, probably almost 3 decades now. So her title, Transforming Growth Factor Beta and Lung Tumorigenesis. Sonia.
Thank you. Well, thank you, Terry, for inviting me to talk to your class today about transforming growth factor beta and lung tumor genesis. And I know that Dr. Moody has spoken to you recently about lung cancer, so I'll start my talk just by highlighting some brief statistics about lung cancer in this country in 2008, the most recent date for which this data is available. Lung cancer has become the most common cause of cancer deaths in the United States among both men and women. In this year, there were more than 100,000 diagnosed new cases of lung cancer and over 150,000 new deaths due to this disease. Most cases now occur in former smokers because of the decline in smoking, the primary cause of lung cancer in this country. But lung cancer is thought to be a problem in the future in other countries because of the increase in smoking, particularly in the Asian countries. And right now, the 5-year survival rate for this disease is still less than a dismal 15%. So this is a very important disease that needs to be studied and needs the people having these problems need help. Now, my lab is interested in transforming growth factor beta, or TGF-beta for short. TGF-beta is a multifunctional regulator of cell proliferation. It's a potent inhibitor of most normal epithelial cells in cultures and shows widespread tissue expression. TGF-beta has been shown to play a pivotal role in maintaining epithelial homeostasis, and it's been shown to be associated with various types of cancers, including human lung cancer. It shows context inhibition or stimulation of cell proliferation and neoplastic transformation, depending on the cell type. So we think TGF-beta is an attractive candidate for new therapeutic intervention approaches in lung cancer. Now, to take you back to the beginning, TGF-beta has its roots in another growth factor called sarcoma growth factor. This growth factor was identified by Joe DeLarco and George Tadaro in the 70s. And it's a polypeptide that was found to be secreted by Maloney-Murine sarcoma virus transformed mouse fibroblasts that stimulates normal rat fibroblasts to form colonies in soft agar. And this was the transformation assay that was used to identify it and follow it. Following this, 2 types of TGF were isolated from these MS3 transformed cells by Anita Roberts and Michael Sporn in the early 80s at the NCI. And one class was found to compete with epidermal growth factor for receptor binding, and this was dubbed TGF-alpha. The second class was found to not be able to compete for EGF binding, but instead had colony-forming activity that was enhanced by EGF, and this was dubbed TGF-beta. So following this, TGF-beta was purified at the NCI from at least 3 sources, including human platelets, human placenta, and bovine kidney. Now, to give you some idea of the scale of this purification from bovine kidney, typically a preparation started with 100 grams of bovine kidney obtained from the slaughterhouse. It's extracted with 8 liters of acid ethanol. Okay. Do I need this? Pardon. Okay, let's continue. It's extracted with 8 liters of a mixture of acid ethanol and then precipitated overnight in the cold room with about 50 liters of ether ethanol mixture. Following this, it is redissolved in 2 liters of acetic acid and then applied to an 80-liter biogel P50 column where 1 liter fractions were collected and processed for further chromatography. And typically, a yield was about 5 to 6 microgram per PARP of purified TGF-beta 1. And to give you some idea of the columns that were used for this purification, this is a typical preparation with using the biogel column. And here is given a 55-gallon drum for reference. Now, the assay for TGF-beta employed the growth of normal rat kidney cells in soft agar. Typically, a mixture of the media, serum, normal rat kidney cells, EGF, and a portion of the sample was plated on agar plates. These were incubated at 37 degrees for 1 week and then stained. And the colonies were counted using an Omnicon image analyzer. So if no colonies were present, if no TGF-beta is present, we wouldn't expect any colonies to be present. If TGF-beta is present in the sample, then there should be colonies that would be counted. So shown on this slide is the final HPLC purification of a typical prep of TGF-beta. And shown in the inset is a polyacrylamide gel showing a 25 kilodalton single band on the gel. This was purified TGF-beta. And Drs. Michael Sporn and Anita Roberts at the NCI are credited for birthing TGF-beta. Now, following the purification of TGF-beta, the amino acid sequence was determined, and TGF-beta was found to be a pre-pro polypeptide of 391 amino acids containing a signal peptide at the N-terminus, a middle portion called the latency-associated peptide, or LAP for short, and a mature TGF-beta at the C-terminus consisting of 112 amino acids. And shown in the inset is the amino acid sequence of TGF-beta, TGF-beta 1 in human. And I will point out the 9 cysteine residues that are typical of TGF-beta. Now, to summarize, TGF-beta is a 25,000 molecular weight disulfide bonded homodimer. It is 3 highly homologous isoforms of TGF-beta exist in mammals. They are called TGF-beta 1, 2, and 3. Principal sources of TGF-beta include the platelets, bone, and spleen. Most normal cells express TGF-beta and its receptors, and TGF-beta is usually secreted in what is called a latent inactive form, which must be activated in order for the molecule to perform its various activities. And the superfamily of TGF-beta contains not only TGF-beta, but the activins, inhibitors, BMPs, and growth differentiation factors. Now, following the amino acid sequence of TGF-beta, the crystal structure of TGF-beta was established at the NCI, and TGF-beta was found to be a dimer of 2 identical monomers that is held together with an interchange disulfide bond and contains a hydrophobic pocket which is needed for interaction with its receptor molecules. Now shown here that is what is called the TGF-beta superfamily. TGF-beta is the protocol member of this family. TGF-beta 1, 2, and 3 exist in the mammals. TGF-beta 4 and TGF-beta 5 have been identified in birds and amphibians and are thought to be the replacements for the mammalian TGF-beta 1. In addition to the TGF-beta, we have the bone morphogenetic proteins and the growth and differentiation factors, as well as the 6 activins, inhibins, and Müllerian inhibitory substance that are part of this superfamily. Now, the TGF-beta superfamily is thought to play a central control role for many biological processes, including such important processes as development, immune system function, reproduction, angiogenesis, aging, tissue repair, metabolic regulation, and homeostasis regulation. There are many processes that are regulated by TGF-beta. Among the most important are that TGF-beta is able to inhibit proliferation of most normal cells, as well as some tumor cells. It also is able to regulate such important processes as apoptosis, differentiation, and immune cell function. It also stimulates the accumulation of extracellular matrix and promotes the process of chemotaxis. Now, a model for how signaling occurs by TGF-beta involves the TGF-beta receptor complex, and this was worked out in the early 90s. So TGF-beta, the ligand shown in blue, binds to the type 2 TGF-beta receptor, which is constitutively phosphorylated. Once this complex is established, it then recruits the type 1 TGF-beta receptor to form a supercomplex, and the type 2 receptor transphosphorylates the type 1 receptor in order for signal transduction to occur and activities such as cell cycle arrest and gene activation and a whole host of other processes may occur. Now, in addition to the receptors, there are receptor-activated and inhibitory SMADs that are involved in this signal transduction process. For TGF-beta and activin, the SMADs 2 and 3 are the principal players that transduce the signal. For the BMP, the SMAD 1, 5, and 8, depending on the particular molecule that is involved. These then transduce the signal to a common SMAD4, which then is able to transport the signal from the cytoplasm into the nucleus. In addition to this, there are inhibitory SMADs, SMAD6 and SMAD7, that can short-circuit the whole process. Now, to put this all together, we have the matrix-bound latent TGF-beta, which is excised from the matrix, and then combines with the type 2 receptor, recruits the type 1 receptor, and then transphosphorylates. Let me show you, transphosphorylates type 1 receptor. Then the SMADs, depending on the molecule, the SMADs 2 or 3 are recruited, and they transduce the signal from the receptor complex to the SMAD proteins. The common SMAD4 then is recruited, and it is the SMAD4 that is able to transduce the signal into the nucleus, whereby it can interact with various transcription factors and cofactors and turn on or turn off key target genes. In addition, SMAD7 is the inhibitory SMAD, which can inhibit the entire process. Now, clinically, TGF-beta has been shown to be a tumor suppressor, and this is based on at least several lines of evidence. I'm giving you 3 lines of evidence here. Germline mutations have been found in various TGF-beta pathway components that cause familial predisposition to some cancers. And for example, this has been shown in SMAD4 in juvenile polyposis syndrome. Secondly, TGF pathway components have been shown to be somatically mutated or deleted in some human cancers. And this includes the type 2 TGF-beta receptor in human non-polyposis colorectal cancer, as well as SMAD4 in pancreatic cancer. Okay? And thirdly, reduced expression of some TGF-beta1 signaling pathway components or overexpression of some pathway inhibitors have been associated with cancer progression. And it's been shown that the type 1 and type 2 TGF-beta are involved here, as well as the inhibitory SPAN7 and another player called SKI, which is able to interact with TGF-beta. Now, in addition, clinically, TGF-beta has also been shown to be a tumor promoter, because TGF-beta has been found to be highly elevated in many advanced types of human cancer and has been shown to correlate either with the metastasis or the prognosis of this disease. And this has been shown in several types of human cancers, including breast, colon, stomach, liver, and et cetera. And shown in the lower right panel is the prostatic adenocarcinoma that has been immunohistochemically stained for TGF-beta1. And TGF-beta1 is found to sit at the interface with the brown staining between the tumor parenchyma and the microenvironment. So, what role does TGF-beta play in carcinogenesis? Is it a hero or is it a villain? Is it a tumor suppressor or a tumor promoter? Well, it turns out it's both. TGF-beta is a proximal effector of the malignant phenotype. It's also a potent growth inhibitor and tumor suppressor. It's also a pro-metastatic factor. Now, how does it do this? Well, a unifying hypothesis has been put forward that has seemed to withstand the test of time, where TGF-beta is able to switch from being a tumor suppressor under normal circumstances to become more of a pro-oncogenic effector during cancer progression. So how does it do this? Well, we have the normal epithelium, and the tumor suppressor activities of TGF-beta can dominate. It can suppress the growth of any abnormal cell. Now, as changes occur in the genetic and epigenetic context as cancer progresses, we have a potential decrease in the responsiveness to TGF-beta. We also have a possible increase in the expression and or activation of this molecule. Oops. So what we have is that TGF-beta, the pro-oncogenic activities start to dominate as cancer progresses to metastases. So the tumor suppressor activities go from being a tumor suppressor to more like a tumor promoter or pro-oncogenic factor. Now, in addition to the SMADs, which we refer to as the SMAD SMAD-dependent pathway because this depends on the role of SMADs in this pathway. There are various SMAD-independent pathways by which TGF-beta can also operate, including the MEK junk pathway, the MEK p38 MAP kinase pathway, the RAS MAP kinase pathway, the RO-ROCK pathway, and the protein phosphorylase 2A S6 kinase pathway. Now, this seems very complicated, and that's part of the problem. So we decided to work on one aspect of this complex problem involving the role of TGF-beta in the RAS-MAP kinase pathway, because we're interested in lung cancer and lung carcinogenesis. And the KRAS proto-oncogene shows an activational mutation in at least 25 to 50% of human lung adenocarcinomas. It's been shown that mutation of even one allele of KRAS increases the appearance of lung lesions in model system. And it's been shown that there is definite crosstalk between the SMAD-dependent pathway and the RAS signaling pathway. Fourthly, activation of the RAS pathway has been shown to be able to modulate TGF-beta signaling through the SMAD pathway. And in vitro studies have shown that TGF-beta 1 can dominate over the mitogenic effects of RAS, but when RAS is activated by mutation, it can clearly override the anti-proliferative effects of TGF-beta 1. Okay? So how can it do this? Well, it can do this by at least 4 different mechanisms that we can think of. There can be a decreased expression or production of the type 2 receptor, which can short circuit the SMAD pathway and lead to other activities that can result in tumor promotion. There can be hyperactivation in the MAP kinase pathway through activated RAS. This again can short circuit the SMAD pathway and pull it away into other activities that lead to tumor promotion. There can be a decrease in either the level of the SMAD proteins or their activity, which again pulls everything into other pathways for tumor promotion. And there can be other changes in what we call the suppressor arm of TGF-beta that can lead away from the SMADs to other pathways that result in tumor promotion. There are probably other activities that can produce this as well, but we're interested mostly in the role of the RAS-MAP kinase pathway in our work. So the broad goal of my laboratory was to determine the role of TGF-beta in the development and malignant transformation of lung epithelial cells. And this work was done in the epithelial carcinogenesis section, which I led, of the Cell and Cancer Biology branch of CCR. We had 3 objectives. The first, to examine the effect of TGF-beta 1 deletion and KRAS mutation alone and in combination on lung tumor incidence and pathology. Secondly, to determine the early events in the development of lung lesions and their progression. And thirdly, to identify potential signal transduction pathway changes that occur with advancing tumorigenesis. So for our work, we employed 4 mouse model systems, and I'll describe briefly each. The AGA mouse system, the C57 Black 6 TGF-beta 1 heterozygous mouse, the AGA Black 6 TGF-beta 1 heterozygous mouse, and the TGF-beta 1 heterozygous KRAS latent activatable mouse. And I'll probably be speaking just het to shorten what I have to tell you. So we asked 2 questions. The lung tumor genesis affects the TGF-beta signaling pathway. And then the reciprocal question, does the TGF-beta signaling pathway affect lung tumor genesis? So we started with the AG mouse model system because this system has been shown to be very susceptible to chemically induced lung tumors. And the lung tumors that develop in a very time-dependent manner and go through the stages of hyperplasia, adenoma, and then carcinoma. And the carcinomas that develop have been shown to be very histologically similar to human lung adenocarcinoma. And the same molecular mutations have been found in both human and mouse lung tumors. For instance, overexpression of the RAS and loss of p53 expression. So we took we took 2-month-old AG mice and injected them with the ethyl carbamate carcinogen, also known as urethane, and then sacrificed them at monthly intervals for up to a year. And shown here is the tumors that were immunohistochemically stained for TGF-beta 1, the type 1 receptor, and the type 2 receptor in 2-month, 4-month, and 8-month samples representing hyperplasia, adenoma, and carcinoma. And we see distinct brown staining for the TGF-beta 1 ligand, as well as the type 1 receptor in all stages of tumor genesis. However, when we look for staining for the type 2 receptor protein, we see decreased staining for this protein at all stages in the tumors in comparison to the type 1 TGF-beta receptor. Now, this is shown more clearly in this picture where we see distinct brown staining for the type 1 receptor in the tumor, as well as in the surrounding normal bronchiolar tissues. Now, in comparison, we look at the immunohistochemical staining. This should be IHC, not ISH. Terry, you didn't revise the slides. Okay, so when we look at the type 2 receptor staining, we see reduced staining in the tumor compared to the type 1 receptor, but normal brown staining in the surrounding normal bronchiolar tissue. So we also looked at the messenger RNAs for the type 1 and type 2 receptor in 5 mouse lung tumor-derived cell lines, and we see various levels of expression of the type 1 receptor message and the type 2 receptor message. Now, I'd like you to draw your attention to the LM1 and PCC4 cell lines. These have been derived from a ethyl carbamate-induced mouse lung tumors, and we show expression of the type 1 receptor, but very low, if any, expression of the type 2 receptor. So this goes in good correlation with what we're seeing in our mouse model system. Now, we also looked at this staining for the type TGF-beta 1 and the 2 receptor in benzpyrene-induced AG mouse lung tumors as well. And we see brown staining in the tumors for TGF-beta 1 and the type 1 receptor, as well as brown staining in the surrounding bronchiolar tissues. When we look at hybridization, in situ hybridization for the corresponding mRNAs, we see distinct expression for TGF-beta 1 as well as the type 1 receptor. However, when we look at the type 2 receptor for TGF-beta, we see very decreased staining in the tumor for the protein as well as for the messenger RNA, while normal brown staining in the surrounding normal tissue. So the type 2 receptor is being affected in this model system. So we see decreased expression of the type 2 receptor, and this correlates with lung tumor formation in our model system. Now, the next question we asked is, does deletion of TGF-beta 1 affect lung tumorigenesis? And for this, we use the C57 Black 6 TGF-beta 1 mouse. The TGF-beta 1 knockout mouse was generated in the early 80s, and it is born and survives. It looks a lot different, smaller, and runtier than its wild-type littermates, but it can be worked with after birth. However, about 21 days after birth, it begins to succumb to a general wasting syndrome where all the organs sort of shut down. So it's not a very good model system for looking at prolonged carcinogenesis. But when working with these mice, it was noted that when the TGF-beta 1 heterozygous mouse, in which only one allele of TGF-beta was deleted instead of two, when these mice were challenged with a liver carcinogen, they showed a response in increased liver tumors in these mice. Now, surprisingly, they also showed increased lung tumorigenesis in these mice as well. So we're interested in lung tumorigenesis, and in order to make the system better for studying lung carcinogenesis, we crossed the C57 Black 6 TGF-beta 1 heterozygous mouse with the AJ mice, which are wild type for TGF-beta 1, to generate the AJ Black 6 mice that had two genotypes in the F1 generation, the heterozygous and the wild type genotypes. Our plan was then to challenge them with carcinogen to generate lung tumors. So this slide shows that when we stain for TGF-beta 1 in the heterozygous mouse for protein as well as RNA, we see decreased expression of the protein as well as the messenger RNA for TGF-beta 1 compared to the wild-type littermates. When we looked at this expression of the mRNAs by northern blotting, we see decreased expression in the heterozygous genotype, as well as with competitive RT-PCR. So reduced expression of TGF-beta 1 protein and message in the heterozygous mice compared to the wild type, as we expected. So our system is working. Next, we injected 2-month-old mice with ethyl carbamate, as we did before, and then sacrificed these mice over a span of 12 months. And shown here on this 3 graph, we show that there's increased tumor incidence and multiplicity and decreased tumor latency in the heterozygous mouse compared to the wild-type littermates. And if you look at hyperplasia and adenoma, it's where we see that there's hyperplasia and adenomas occurring by 1 month after challenge with the carcinogen. It's not until 2 months and 4 months that we begin to be seeing hyperplasia and adenomas in the wild-type mice. And most dramatically, we see carcinomas appearing by 4 months in the heterozygous mice, whereas it takes at least 12 months or longer before we see tumors appearing in the wild-type mice. Now, we stain these tumors for TGF-beta 1 in both strains, and we see staining for TGF-beta 1 in the wild-type and heterozygous mouse in hyperplasia and adenomas. But however, when we get to the carcinoma advanced stage, the expression of the type 2 receptor for TGF-beta is very reduced in the heterozygous mice compared to the wild-type mice. We looked at the corresponding mRNA for this as well, and we see decreasing levels of the messenger RNA for the type 2 receptor with increasing lung tumorigenesis as we proceed from hyperplasia to adenoma and onto carcinoma. Next, we ask the question, does deletion of TGF-beta 1 and mutation of KRAS in combination affect lung tumorigenesis? And for this, we use the TGF-beta 1 heterozygous KRAS latent activatable mouse. So to study the interplay of TGF-beta 1 with KRAS, we generated mice by crossing TGF-beta 1 heterozygous mouse in mice in the C57 black 6 strain with KRAS latent activatable mouse in the SV129 strain. We obtained these mice from Tyler Jacks at MIT, and they contain what is called a latent activatable mutation, where the KRAS mutation is there, but it requires another event to activate the mutation and get tumorigenesis going. We cross these mice and we generate 4 different genotypes. The TGF-beta 1 heterozygous latent activatable KRAS double mutant mouse, the KRAS single mutant mouse, the TGF-beta 1 heterozygous single mutant mouse, and the wild type mouse as a control. Now shown here are some photographs of the resulting tumors that we found in these lungs at 3 months after challenge with carcinogens. In the double mutant mice, we see pearly nodules on the surface, which are the tumors, and this we see as well in the single mutant KRAS mice. In the TGF-beta 1 heterozygous and the wild-type mice, we only see only a very rare, if any, tumors appearing even after prolonged time. We looked at the effect of TGF-beta 1 gene deletion and KRAS mutation on mouse survival, and of these 4 different genotypes, and showed that the TGF-beta 1 KRAS double mutant and the KRAS single mutant had decreased lifespans in comparison to the TGF-beta 1 heterozygous mouse and the wild-type mice. Next, we look at the pathology of some of these lung lesions, and we see increased numbers of hyperplasia and adenomas in the single KRAS mutant mouse, and increased numbers of carcinomas in double mutant mice. Next, we performed immunohistochemical staining to look at these TGF-beta 1 and type 2 receptor in double mutant adenomas and carcinomas. We see staining for both proteins in the adenomas. However, when we get to the advanced carcinoma stage, in the double mutant mice, we see very reduced staining for the TGF-beta 1 ligand protein, as well as the protein for the type 2 receptor in the adenocarcinomas. And next, we decided to look at the protein levels of some of the TGF-beta signaling components in these mice, and I'll show you a few slides of our results. When we look at the type 2 receptor in the double mutant mice compared to the single KRAS mutant mice, we see an expedited reduction in the level of type 2 receptor protein compared at 2 months compared to 3 months in the single mutant KRAS mice. And we see virtually no changes in the other two genotypes. When we look at the SMAD3 protein, which is a receptor SMAD, we see as well an increase in the time at which we see increased production of the SMAD3 protein in the double mutant mouse compared to the single KRAS mutant mice. So there's an expedited increase in the SMAD3 protein and an expedited reduction in the type 2 receptor protein. Next, we looked at the common SMAD4, and in both KRAS double mutants and single mutants, there seems to be a reduced expression of the type 4 protein, particularly in the double mutant mouse compared to the single mutant mouse. We looked at SMAD7, and surprisingly, very low, if anything, in both genotypes compared to clear levels of the inhibitory SMAD protein in the wild type and the TGF-beta heterozygous mouse. So this implies that there's really no inhibitory protein to counteract this system with the KRAS mice. Next, we look at the corresponding mRNAs for some of the SMADs and the type 2 receptor. Essentially, we find reduced amounts of RNA for the SMADs in the adenomas and reduced as well as in some of the carcinomas, mirroring at the protein level. Since we're interested in the KRAS system, we looked at the expression of KRAS and RAF1, and in the double mutant mouse compared to the wild type latent activatable mouse, we see an expedited increase in the amount of KRAS and RAF protein by about a month compared to the single mutant KRAS mouse. The wild type, we don't see any change with. Since TGF-beta is very important in apoptosis, we looked at the level of apoptosis in these mice, and we see reduced levels of apoptosis in the double mutant KRAS mice compared to the single mutant KRAS mouse. This is also reflected when we do tunnel assay for apoptosis. In the double mutant mouse, we see decreased, reduced levels of apoptosis staining in the adenomas and hyperplasias compared to the single mutant KRAS mouse. And virtually, we can't detect really apoptosis in the advanced carcinoma stage. So putting this all together, in the single mutant KRAS mice, we have the TGF-beta SMAD inhibitory pathway functioning and acting to try to decrease carcinogenesis. But at the same time, we have KRAS, which is a bully, and is able to push the progression of what is occurring because it is such an active molecule. And it's able to, we think, divert a lot of the SMAD-dependent activity into SMAD-independent activity in order to accelerate tumorigenesis. But in contrast, with the TGF-beta1 heterozygous KRAS double mutant mouse, we have the disadvantage that the TGF-beta SMAD-dependent inhibitory pathway is debilitated because there's loss of one TGF-beta1 allele. In combination of that, we have the KRAS, activated KRAS, forcing the same SMAD-independent pathway effects, which lead to carcinogenesis. In addition, we've shown that in our study, there is very much reduced levels of SMAD4 in these mice as well. So this also helps to subvert the inhibitory pathway and to encourage the pro-oncogenic effect of TGF-beta in these mice. So we think these mouse model systems are the way to go to try to understand what's happening to this dreaded disease. And I think Tyler Jackson and many other people have now constructed better KRAS mouse models where you can look at these for longer times and over time periods to really find out what is happening at the molecular level. So in our mouse model system, we're showing that we see decreased levels of the type 2 receptor for TGF-beta correlating with lung tumor promotion. We also see activated RAS-MAP kinase pathway that also correlates with lung tumor promotion. Our system shows decreased levels of SMAD4 in our system, and that seems to also correlate with lung tumor promotion. And finally, compromised apoptosis in our model system also correlates with lung tumor promotion. So I'd like to credit the people who contributed to this work that have since moved on to bigger and better positions. Jerry Anderson, a pre-doc in my lab, Yang Kang, Elena Normova, Justin Pandy, and Sarah Umfers, postdocs. And again, I credit Tyler Jacks for the KRAS latent activatable mouse. And now I'll be happy to answer any questions if you have any and time permits. Okay, thank you.
So are there any antagonists for the TGF-beta receptor?
Their antagonists are being developed, okay? Unfortunately, they're very difficult to produce, and they showed minimal effects on lung cancer itself, but they're in the works. Have I overwhelmed you? Okay. Well, thank you. Thank you so much.